everybody. Welcome to the second week of Fall Quarter Quantitative Seminar. My name is Abby Bratt, and I'm one of the organizers of this year's Quantitative Seminar, along with my colleagues Amelia Duval and Brielle Thompson. And I'll begin by acknowledging the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. And we also acknowledge that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. So Curtis asked for a short introduction and to be introduced as such. Curtis Deutsch is an earth scientist studying the interactions between climate and ecosystems. With his students and colleagues, he combines mathematical models of varying complexity with diverse streams of biological and physical data to discover the ways in which climate produces spatial pattern and temporal variability in ecosystems. Most of this work has focused on how the ocean produces, transports, and recycles the nutrients and oxygen that sustains its plant, animal, and microbial ecosystems over a range of timescales from years to millennia. He also works with terrestrial ecologists to understand how climate influences the patterns of thermal fitness and their implications for biodiversity and biogeography in a changing climate. He is a fellow of the Kavli Frontiers of Science and received the Rosenstiel Award from the University of Miami and an Investigator Award from the Gordon and Betty Moore Foundation. He has previously held faculty positions at UCLA and the University of Washington and is now a professor in the Department of Geosciences at Princeton University. So with that, take it away, Curtis.
Thanks very much. I wish that I could see all of your faces, many familiar and some unfamiliar, and that they would all be smiling at least through the first 5 slides. Anyway, here we will go. I want to talk about climate change and the body size of marine species. And this is work that I started while I was at University of Washington. And so some of my former students and postdocs and current students and postdocs are listed down there along with funding agencies. But let me just jump right into the thick of it. It's something that everyone knows, which is that body size is really important, and it's important to all kinds of processes in the Earth system, you might say, starting from physiology. So on the left, it's a really wonderful study compiling metabolic rate on the y-axis versus body size over many, many orders of magnitude, from unicells down here up to large metazoans up here. And you see that approximately linear or piecewise linear relationship between metabolic rate and body size that's kind of famous or infamous. And so there's at an organismal scale the importance of body size influencing metabolism, and I'm going to come back to that later in the talk. But of course, at higher orders of organization in biology, one sees body size playing an important role in, say, the abundance of populations. This is for some terrestrial mammals, just showing that as body size goes up, the abundance goes down, again, in a very power law fashion, linear on a log-log plot. And then finally, at sort of planetary scales, if you think about biogeochemical cycling of carbon, say, in the global ocean, there's a really important role for body size in governing the particulate matter that ultimately buries carbon in the deep sea, sustains lower atmospheric CO2 and oxygen in the atmosphere. And those particulate volumes from, in this case, some copepods, varies very strongly with the size of the copepod. So at all the levels of biological organization on Earth, you see these really strong relationships to body size. But of course, body size itself can be influenced by changes in the Earth system. And so here's one rather nice example from a study across the Eocene-Oligocene transition. It's a period of cooling that's shown in the blue curve here. And along with that, the fossils of ostracods show a pretty coherent kind of 2-step increase in the size of the fossils. And so there's our first indication that something in the climate system that was causing the Earth to cool in this kind of stepwise fashion was also causing, well, one presumes, an increase in the body size of animals. Now, of course, nature is infinitely complex, and although the temperature is one measure of how the climate was changing, it's not obvious from this picture that it was temperature that was the ultimate cause. It could have been an indirect cause of that. So if we want to know why the climate change drove body sizes to vary, not only among ostracods, but among lots of different taxa in the fossil record and different time intervals, we of course turn to the a mainstay of science, the reductive nature of science to find the root causes of things, and for that we would like to seek out laboratory experiments, and those were really nicely compiled by a series of authors going back about a decade now, in which every single point on this plot is an experiment with a given species, and the species are all aquatic species here, but in the triangles at least, there are some terrestrial metazoans in the circles. And what's been quantified is the magnitude of body size change per degree of warming. Now, the way that's done is in so-called rearing experiments, where organisms are harvested from their natural environment, they're brought into a lab, they're grown under a certain temperature, and then the progeny of those reproducing populations is moved into a different water bath at a different temperature and allowed to grow to maturation, and the adult size or maximum size of that population is recorded, and that size, as you see here, tends to be smaller for every species that's below the line. So this line would be 0 change in body size per degree of warmer temperature. Almost all the species fall below the line, which means that on average, most species are getting smaller as temperature goes up, consistent with what that paleo record of fossils in ostracods also showed. There are some exceptions to that. There are some species that actually get larger as you warm up their environment. But at least among marine species, it's the vast majority get smaller. And there's also, as these authors pointed out, a hint that as you, by the way, body size here is on the x-axis. As you get toward larger and larger body size, of course, this is a log 10 scale. So this is an enormous, from ciliates down here up to larger zooplankton up here, about 1 gram in size. As you increase in that body size, you start to see larger and larger responses of body mass to a given degree of temperature. You just take, among the kind of metazoan populations, leaving the unit cells behind here, you just take a kind of average of these triangles for the aquatic organisms. You get something like a 4% change, reduction in body size per degree. Okay, so there's a simple number to keep in your head for a while. But there's lots of questions raised by this, like what is the mechanism? Why physiologically do things get smaller when you subject them to warmer environmental conditions? If you take at face value that among aquatic organisms that tendency to get smaller actually increases as you get toward larger size, what would be the mechanism for that? Why does it seem to be a stronger response for larger species? And really, how far does it go? So, these experimental results stop at 10 to the 3rd milligrams, 1 milligram body size. Does it go all the way up to the things that folks in fisheries would typically be concerned about, or people standing on the beach looking at the fauna of the ocean? Does this so-called temperature size rule extend to those larger organisms for which direct experimental evidence is lacking or very scarce? So that's the kind of question, all these questions indeed, are the kinds of questions for which one might turn to a model. And so that was, I think, first done in this paper about a decade ago. It's a very provocative model. It starts from a very common description of body size growth in fishes, the von Bertalanffy curve, which has just a few parameters, and those parameters are typically derived empirically from measuring growth with age of individual species. And if you assign some reasonable kinds of temperature dependencies to those, to the parameters in the von Bertalanffy curve, one can make an estimate of, a model-based estimate of how large the size change would be in a climate scenario of the future in which the oceans have gotten warmer and oxygen has gone down critically, because, forgot to mention in this past slide, one of the key hypotheses for why this happens is that as you get warmer, metabolic demand for oxygen goes up, and if oxygen in the environment doesn't go up to keep up with that increased metabolism, one sensible way to increase your oxygen supply relative demand is to make your surface area bigger relative to volume, which can be done by getting smaller. So there's an underlying hypothesis for these trends that was sort of formalized in a semi-empirical model based on the von Bertalanffy curves here. And it produced this rather provocative result. There's a whole lot of variation you see on the map. You know, some things get much smaller in the reds and yellows. Some things actually get bigger in the blues and turquoise. There's a whole lot of spatial structure here. It's not entirely clear where that comes from, but the most important aspect of this model was that it provoked a very vociferous debate. Some of you, I think, have been involved, at least peripherally, in that debate, if not centrally to that debate, about shrinking fishes. Part of the reason that it generated so much debate is that the connection to the underlying physiology of oxygen limitation was not the main plank of this model. It was built on a kind of semi-empirical set of curves, and so naturally, folks who think a lot about physiology and oxygen and the limitation of growth by oxygen had things to say about whether that was a realistic representation. And so a debate ensued, primarily playing out in what you might call, or what the authors and editors called, the opinion pages of major scientific journals, arguing about whether growth is indeed oxygen limited and therefore would come to play a role in shrinking fishes of a warmer ocean. So there's, roughly speaking, we could say there are sort of two camps. One that says yes, rising temperatures and lower oxygens will act to make animals mature at a smaller size, and others who say no. And the basic argument comes down to this. On the one side, you have a camp that says, well, as body size increases, metabolism, metabolic rate per individual grows, and if the area presented to the environment for extracting oxygen doesn't grow enough to keep the oxygen supply matching the demand, then you'll run into oxygen limitation, the only solution to which is to stop growing at a smaller size. Okay, so it's kind of a geometric argument underlying this. Oops. The contrary opinion was that well, though the area for extracting oxygen from the environment doesn't grow isometrically with body size, you know, it occurs at gills, and gills are highly folded surfaces, and their area can grow with size in any way that suits the selective pressures acting on the organism. And so the idea was that as body size grows, the area will just grow to match whatever the metabolic demands of a given species are. Okay, so this is the debate as it's played out in these papers. Some of you will know it quite well. One thing you'll immediately recognize is that although the underlying hypothesis here is about oxygen limitation, the debate centers on geometric arguments, areas of gill and how they increase with body size, rates of metabolism and how they increase with body size. Notably missing from the arguments on either side just to a large degree, is measures of actually how measured oxygen limitation varies with body size. Now that's a generalization. There have been some data presented to support each camp, actually, but generally speaking, these arguments have been advanced on the basis of geometric scaling of supply and demand. So what I'd like to do is take a different approach to this same question and ask whether we can start from basic physiological principles to derive a mechanistic model that predicts the magnitude of those temperature size responses, the effective temperature on body size that we saw in the previous slide, from those more fundamental measured physiological traits that we get from laboratory experiments. And for those of you who can't stick around to the end of the talk, spoiler, the answer is yes, we can do that. Whether the model reconciles those two divergent views is yet to be seen, of course. I would like to advance the answer that yes, it can. Maybe not completely, but it's at least a step toward reconciling that debate. So that's my take home for those who can't stick around. Now let's dive into the nuts and bolts of this mechanistic model. And I'm going to go way back to something hopefully everyone has studied. I consider it a kind of seminal paper in ocean science from Fry and Hart, just sort of a series of papers in the late 40s, first presenting a variety of measurements about how, in this case, goldfish respond to different oxygen tensions and temperatures in terms of their oxygen consumption rate. So this is our metabolic rate here, right? Measure of how much oxygen is consumed per gram of body mass per unit of time. And you can see that if you're at high enough oxygens, that rate is pretty independent of time, but it's, independent of oxygen, strongly dependent on temperature. And it rises pretty quickly as you go from cold temperatures up to very warm temperatures. That rate of oxygen consumption goes up. No surprise, that's been known since even before Fry and Hart, going back to the early 20th century. And we can characterize that demand through a very simple expression that says for any given species, there's some sort of reference level demand at a given temperature and body size. But of course, that demand will increase with temperature according to this Arrhenius function. It's just an exponential function of temperature, has slight peculiarities because temperature is in the denominator, but don't worry about that. Think of this as just exponential increase with temperature, and it can also increase with body size, as we know it does. You know, there's 1,000 papers written on this exponent delta, which is canonically something like 3/4. Yeah. Okay. So there's an expression for demand which works as long as the oxygen is high enough and demand is not limited by oxygen. But of course, as you go toward oxygens that are low on the left end, that oxygen consumption begins to go down with oxygen depletion. And that is sensibly taken as a reflection of the fact that oxygen is getting so low it can't support that demand anymore. And what we're really seeing is the rate at which oxygen supply to the animal is decreasing as oxygen goes down. So that gives us another simple expression and another simple curve, which is not the flat curve, but the linear curve coming from the origin, rising with oxygen in the environment. That's this oxygen tension here, oxygen pressure. And you could say that the supply rate of oxygen also has some kind of species-specific exchange coefficient at a reference temperature and body size, and it increases the overall supply, will increase with the ambient pressure of oxygen in the water. So that's just the expression for this line right here. Of course, this reference coefficient, which is the slope of that line, can vary with temperature too, and we see that in these experiments, right? As temperature goes up, the slope of this curve goes up from a relatively shallow slope at low temperatures towards steeper and steeper slopes as we get toward the higher temperatures. What's not shown here, but you can imagine, is that that slope will also vary with the size of the animal. And one critical parameter for us is going to be this sigma, the rate at which the oxygen, or the degree to which the oxygen supply increases for a given increase in the body size of the animal. That's not shown in these experiments. We're going to need to get that in other ways. Okay. Let me move to the next thing, which is if we take the ratio of oxygen supply, that potential supply, this curve here, it's potential because it is really the oxygen supply at low oxygen, but it continues to rise at high oxygen, and it might not, all that potential oxygen supply might not actually be realized because it's not being matched by consumption. If we think about the ratio of potential supply to that resting metabolic demand that we saw before, we get a simple expression, just taking the ratio of those two things. And it's going to, for us here, define a parameter I'm going to call phi, or the metabolic index. It's just the ratio of potential oxygen supply to resting demand. And it's got a coefficient here, which is the ratio of the two previously noted coefficients, the supply to demand ratio for a reference temperature and body size. It can vary with body size now according to epsilon, which is just the difference between the allometric exponent for supply variations and the allometric exponent for demand, metabolic rate variations with size. It will increase linearly with the ambient O2, and it can increase exponentially with temperature. Okay, so if the rate at which demand increases with temperature, that is the height of the flat parts of the curve, increases faster than the slope of those oxygen limited portions of the curve. Okay, so let's look at how that plays out. Lost my cursor. Okay, here we go. So now we can just do what everyone wants to do with a supply curve and a demand curve. You find the market clearing equilibrium price. You just put the curves down and you find where they cross. The price, of course, is really the oxygen pressure in the environment in this case that allows supply and demand to exactly balance one another. So that is given a name in the physiological literature. It's called P-crit, the critical partial pressure of oxygen. And you can observe that by just any one of these experiments where oxygen reductions don't change metabolic consumption of oxygen until the point at which that P curve is crossed, and then the oxygen consumption rate follows the supply, not the intrinsic demand, and the knee in that bent curve is the P-crit value. Now, if you put P-crit into our expression for the supply to demand ratio at a variety of temperatures, you know that phi at that point, the potential supply and demand, resting demand, are equal, so you can set that equal to 1. And interestingly, if you do that at multiple temperatures, you can get the key parameters, the E0, the temperature sensitivity of the oxygen supply-demand ratio, i.e. the metabolic index. If you do it at multiple body sizes, you get this other exponent, epsilon, the degree to which body size influences the break-even pressure of oxygen that allows supply and demand to be balanced. Okay. So there's a lot of variables floating around here, and I'm sorry for that. I just wanted to get them on the board, and I'll try to keep reminding you what they are as we go through and use them. But the two that you really want to pay attention to are epsilon, which governs how the oxygen supply-demand ratio varies with body size, and E0, the exponent that describes how the oxygen supply-demand ratio varies with temperature, because after all, we're interested in how temperature influences body size, and those are going to be the two obvious key parameters, E0 and epsilon. Okay. Got to say one more thing though about this, which is that everything so far has been data from a resting rate of metabolism, but of course in nature, species and organisms need energy above that resting rate in order to do all kinds of ecological activity. So if you're measuring metabolic rate, the minimum rate you'll measure is the resting rate, but at the maximum rate you might measure would be a maximum metabolic rate, the very fastest rate at which energy can be metabolically transformed in a given species. And somewhere in between is going to be a sustained rate that's needed to stay alive in an ecosystem, but wouldn't be enough to do maximum exertion at all times. Okay, so each one of those two relevant thresholds, a resting rate, a maximum rate, and something in between that you need to be able to sustain over long time scales to maintain a viable population, they'll each have associated with them a different oxygen threshold. So obviously the resting rate has that threshold of P crit. The partial pressure of oxygen has to be greater than P crit to sustain resting metabolism. That corresponds also to a minimum value of 5.1. If you want to sustain maximum exertion indefinitely, you'd better multiply that P crit in the environment by the ratio of your maximum metabolic rate relative to the resting rate. That's going to require oxygen in the environment to be that factor higher than what was needed in the resting state. And that will require phi in the environment to be also higher than 1 by this number, phi max. And if you can survive long-term on some kind of sustained metabolic rate that's between resting and maximum, you could get away, you could survive with a phi of something that I'm going to call phi crit. Okay. And this is also shown in a wonderful, even earlier paper by Frey in 1947. Okay, so here's the model of the temperature size effect. Let's just put some contours of phi on a plane that is that has temperature on the x-axis and body size on the y-axis, log 10. Phi contours for most organisms, as we'll see in a moment, would slope down to the right, where low phi would occur at high temperature or large body size, and high phi would be down here. So this is sort of moving toward oxygen limitation up here and non-oxygen limitation down here. If you imagine a species that sits on some contour of the metabolic index that is already limiting its growth or activity, and you increase the temperature, its phi is going to decline. I've lost my cursor. According to this red line, and the magnitude of that, the length of that red line can be calculated as the amount of warming that you apply, but multiplied by the temperature sensitivity of the metabolic index of the hypoxia tolerance, if you want, or the oxygen supply demand ratio, divided by Boltzmann's constant times the square of the temperature. This is basically a constant. Okay, so the reduction in your oxygen supply demand ratio is proportional to the temperature change that you experience and your temperature sensitivity. Yeah. If you want, that's going to put you on a different phi contour. If you want to get back to the original one and you can't control the temperature, you might be able to control your body size, or at least your progeny's body size. And if you can do that, make your progeny get smaller, they can come back to the original ratio of oxygen supply to demand, the original fitness associated with a warmer environment, by just getting smaller. And of course, the length of that blue arrow we could just calculate from the definition of the metabolic index, and it's proportional to the fractional change in body size and proportional to that allometric exponent epsilon. Okay, so here's our two key parameters: epsilon, the allometric exponent for hypoxia tolerance, and E naught, the temperature sensitivity of that hypoxia tolerance. If we just want these two arrows to be equal, we get a very simple expression for the fractional change in body size per degree of warming, which defines, pardon me, the temperature size effect, okay, that all those experiments measured way back on slide 3. And that temperature size effect, we ought to be able to predict from the ratio of the temperature sensitivity of hypoxia tolerance, E naught, relative to the allometric exponent, epsilon, okay, and then these other constants. That's, of course, assuming that the organisms start off in an oxygen limiting scenario, but of course, maybe that isn't true. You might want to think about organisms that initially have some kind of excess of oxygen, and they start off at an initial phi here that allows them to reduce their oxygen supply demand ratio without getting smaller. Now, there could be an energetic cost to that. They may have to reduce activity to move from this high phi down to this lower phi, but that may be okay as long as they haven't crossed the sustained threshold for a population's survival. If they go further than that, of course, then they're going to be in trouble. If we think about an organism that has this, what I'm going to call an activity buffer, or an aerobic activity buffer, this little stretch of orange vector here is what you can tolerate without getting smaller. It turns out you just go through the math, and it just is a very straightforward modification of what you predict the temperature size effect to be. You just start with your previous expression, and you just multiply by 1 minus f. That is the fraction of your metabolic index reduction that can only be compensated by size reductions after you've exhausted all other possible adaptations like reduced activity, okay? All right. What this means is that if we can estimate the activity buffer for a variety of organisms, the E naught for them and the epsilon, then we could predict what the temperature size effect would be from pretty fundamental parameters that, you know, don't already tell us what the temperature size effect is. We can use those physiological traits to predict it. Okay, so that's what I want to do. The good news is we have lots of data. The data is comes from decades of, you know, people making measurements in a lab that are the beneficiaries of, have gone through the literature and pulled out that data. Lots of it on temperature-dependent hypoxia traits, metabolism, and P-crit values across a range of temperatures. And also, of course, lots of experimental data on the temperature size effect. I've already shown you that. So for now, we can at least statistically test whether the predictions of the temperature size effect are realistic. The bad news is that the species for which we have all those parameters together is basically the empty set. We don't have those same parameters for the same species, but we have lots of different species, and we can draw histograms of the frequency of those traits. So that's what we're going to do. And we want to compare it, go back to the original observations for TSE, and we want to compare statistically, can we reconstruct this distribution of temperature size effects from the more fundamental physiological traits. So you've already seen the underlying data. That here I'm just plotting plotting it in a histogram fashion. Okay, so again, you may remember the first number I quoted to you was about 4 degrees. 4% reduction in body size per degree of warming. That's just the median of, lost my cursor, the median of this histogram. But not only would we like to predict the median, we'd like to predict the overall shape and variance of that histogram from the fundamental traits. So this is the curve we're going to use to test whether oxygen limitation can explain this data. Okay. So the first thing we need to do is grab a bunch of traits that describe temperature-dependent hypoxia, so the E naught values, okay? And we've done that for a variety of different purposes. Here I'm just reusing it for this body size study. The data comes from, you know, a large number of species, a tiny fraction, of course, of the ocean's species, but a large number of laboratory studies have been needed to get us something getting close to 100 different species that come from all ocean basins, equator to pole. They span the whole range of oxygen and temperature conditions in the modern ocean. Several different phyla. There's, you know, a sampling of the different organisms that we have these traits from. And here's what the data looks like. Put all those species on a temperature versus natural log of the P-crit oxygen threshold. Most of them slope up to the right. That is, they require a higher oxygen in the ambient water to meet resting metabolic needs if the temperature is warmer. And the slope of that basically gives us a histogram of E naught, here measured in electron volts. And that's, for those of you who follow the metabolic theory of ecology, you know, these numbers are similar to what you get for metabolic rate, but actually a bit lower, which is an indication that oxygen supply is responding to temperature to compensate part of the increased oxygen demand. In any case, this just is what it is for now. For whatever reasons, E naught has this histogram. And from that, we can actually make the first step toward predicting what the temperature size effect would be, just ignoring these other parameters for the moment, and seeing if we just take E naught over KT squared, how big is the reduction in aerobic scope in metabolic index as a % per degree. And here we get our first kind of sensible histogram. It's centered on about 4 degrees per degree of warming. 4% per degree of warming with a pretty wide range, okay? Already it looks like we might be within striking range of predicting the magnitude and variance of the TSE, but of course we need to deal with these other parameters, and they turn out to be much harder. Okay. Remember that the activity buffer is sort of how much loss of the metabolic index can be sort of accommodated by just reducing activity before basic population survival is threatened. And we can estimate that by looking into the biogeography of species and asking across their whole range, by how much does the maximum metabolic index that they inhabit exceed the lowest metabolic index that they inhabit, with the idea that this lowest metabolic index that they inhabit, phi crit, tells us something about the requirement for sustained energetic extraction from the environment, and that this phi max is sort of at the upper end of the species range. It's sort of how much they could tolerate moving to the other end of their geographic range before that limit is hit. So if we look at the maximum phi and the minimum phi, what we call phi crit, within the geographic range of species, we can estimate that activity buffer. How much phi can they reduce before they're basically excluded from the environment? Okay, so we've done that for other reasons again in other studies. Here's just a couple of highlights from that. The species geographic range of different species with different traits are shown here. There's a tropical species confined to shallow water near the tropics by a metabolic index that actually decreases toward the pole and decreases strongly with depth. And you can see other very different ecotypes with very different biogeographies. For example, one that's confined to high latitudes, excluded from the low latitudes by the combination of temperature-dependent hypoxia providing too little energy to stay alive. The trade-off, of course, is that they can actually live at greater depths. Part of what excludes them from the tropics is that their hypoxia tolerance gets their hypoxia thresholds get higher as temperature warms, and that means they can actually exploit habitat to deeper depths. The point of this is really that we can find a minimum phi that corresponds pretty closely to the boundary of the geographic range of species for a wide range of different species. Turns out that value, phi crit, does a better job of delineating habitat than either temperature or oxygen does on its own, and so we have reasons to think that this phi crit is actually represents an aerobic limit to habitability. There are some other indications that that's true that I'm just going to skip for now in the interest of time that we can talk about afterwards for those who are interested. The key thing is now with that lower limit that represents an aerobic energy limit to habitability, we can compare that on the y-axis here to the maximum value that's inhabited, and you find that that species inhabit about twice, they inhabit a range of phi that's about twofold when averaged across species with a fairly narrow range. So we can use this data to estimate that activity buffer, and for most species, by the time from your maximum metabolic index down to the minimum that you inhabit, there's about a factor of 2 reduction in the potential aerobic scope, or that potential oxygen supply to demand ratio. If we look at that across all the different species where we can estimate it, again, it's a distribution centered on about a half, but there's no discernible relationship to body size. Okay, so now we can just put these pieces together. We need one more piece, which is the allometric exponent that regulates the oxygen supply to demand variation with body size. We can do that in much the same way that we did for temperature, except that we're using now experiments that didn't vary temperature, but varied instead the body size. I'm not going to go through all those experimental data. This is just a summary of them as a histogram of each species epsilon value. And here you can see an interesting thing. First of all, on average, the values are negative, which means that as body size increases, the oxygen threshold for physiological survival increases. That means the critical oxygen pressure rises as body size goes up. Things become less hypoxia tolerant as they get bigger, on average, according to the data that exists in the literature. It does not exclude things that have no size dependence, that don't care whether they get what oxygen is as they get larger or smaller. A 0 would mean they're independent, their hypoxia tolerance is independent of size. So there are plenty of species that are within that realm too. The other interesting thing you can see here is that there does seem to be a correspondence with body size. So I've colored these histograms by body size, and the smallest ones have the biggest allometric sensitivity for their hypoxia tolerance, and the largest ones are clustered closer to 0. Okay, so already you can see that there's something interesting maybe going on with size here. But for now, I'm just going to set that aside and ask, if we just ignore the variations of epsilon with size, can we reconstruct the temperature size effect as we set out to do? So here are the different component parts. We have the E naught over KT squared. We have our activity buffer, which is around half. And then we have our epsilon, the allometric scaling of hypoxia tolerance, where at smallish sizes, we get something around -0.4, -0.3 for the size of organisms that we have temperature size experiments for. And when we put those together, we predict a distribution, a mean and variance of the temperature size effect that's in the blue bars. And it matches up reasonably well, in fact, maybe strikingly well, to the observed curve that I showed you before from direct experimental evidence from the rearing experiments. So it appears that using basic physiology of oxygen limitation and traits that come from laboratory studies, we can predict independently what the temperature size effect mean and distribution should be. And it does a reasonably good job. But there's this remaining issue of how it varies with size that I want to come back to. And again, just to remind you, the allometric exponent seems to get closer to 0, meaning that things become less sensitive to their hypoxia tolerance. It becomes less sensitive to size as they get toward bigger, as we get toward bigger species. At the same time, the magnitude of the temperature size effect is very subtle, but it seems to get a little bit bigger as we get toward bigger sizes, right? So these dark blue ones here are the largest sizes in the database of rearing experiments, and they tend to lie out on the flanks here, whereas the smallest sizes of species tend to be on the low end of the temperature size effect. And that's exactly what we would predict from the equation, which I should have included again here, right? As epsilon gets smaller, we would expect temperature size effect to get bigger, and indeed it seems to, but can we do better than that in 4 minutes? I doubt it, but I'll try anyway. Here's some data on metabolic rate. One of the slides I started with, just showing the metabolic rate versus body size in red points across all taxa that have been measured, terrestrial and marine. And if you just fit a curve to that, you can get the exponent as the slope of that curve in log space. And you find that the allometric scaling of metabolic rate starts off greater than the canonical 3/4 when you're in the unicellular size range for reasons that aren't entirely clear. And they get down toward that canonical 3/4 power scaling as you get toward the large end. And in fact, that phylogenetic variation by just throwing all species together gives you a number that's pretty close to what people get when they look at ontogenetic variation, which has a wide range, but it's around 0.8 or so. Okay, so there's a curve that describes the scaling of metabolic demand for oxygen. What about supply? Supply could be a whole other seminar, but I'm just going to point out that we have a theoretical model that starts with theoretical boundary layer diffusive scaling, and that predicts that sigma should be 1/3. You might think 2/3 at first, but remember that the thickness of the boundary layer also increases as an object gets bigger, so that kind of knocks sigma down from 2/3 to 1/3, And then on the other hand, we don't have theory, but we have a variety of empirical studies that suggest sigma should be around 0.7. We can actually reproduce the theoretical limit and the empirical limit with a fairly simple semi-theoretical, semi-empirical model, and I can't go into the details, except that it reaches both those limits rather nicely. And if we take the difference here between the demand allometric exponent and the supply allometric exponent, we can now focus on the green curve. So you've seen these top two panels. Here's the green curve, which is epsilon, that size dependence of hypoxia tolerance across a huge size range from unicells all the way up to 100-gram animals. Okay, and the estimates of epsilon are in the points from each of the individual species. You've seen that in a histogram before, and the theoretical curve is in the green. Okay, so now this theoretical curve, it seems to fit the data pretty well. We had one free parameter to play with, so that helps. But we can take the slope of that green curve, which is epsilon versus body size, and we can ask whether it reproduces something like the body size dependence of the body size response to temperature. So here's that again, log body size on the x-axis, the temperature size effect, the response to a degree of temperature warming on the y-axis. And we saw before that the data seem to hint at a stronger response among larger size organisms. And the green curve now, based on that theoretical expression, that theoretical model of oxygen supply guided by empirical traits, reproduces that. Okay. So let me just take my last minute here and go back to thinking about the future, which is what motivated a lot of this in the beginning. And we can generalize the expression for the body size, for the temperature size effect to include not only temperature changes, but also oxygen changes. The math is pretty simple for that. It just comes right along for the ride. When you have a temperature, an oxygen pressure that's declining as temperature goes up, which is what most of the ocean does. And from that, you can then just compute what is the fractional change in body size for given traits typical of, let's say, in this case, zooplankton at the base of the marine food web. So the for the warming and oxygen loss of the projected for the upper 500 meters, excuse me, over the century, a body size of 1 gram at the upper end of the range of observed temperature size effects. We use the traits here, and we get, something like a 20 to 30% reduction on average, about 20% on global average in the body size for things if they can't move to escape a lower oxygen supply and can't adapt their traits fast enough. So those are two key caveats here. We assume that there's no other response than just body size changes. We would get something like 20 to 30% over large parts of the ocean. Of course, that depends very strongly on the traits that you choose, right? It can be virtually zero response if your temperature sensitivity to of hypoxia is very low, or if your allometric exponent is very large. Interestingly, as the allometric exponent gets closer and closer to zero, as people have argued for folded gills, actually the temperature size response, the TSE, gets bigger, not smaller. And that's illustrated here, my last slide, just thinking about how we would use this to think about extinctions in the past, for example, since that's one of the very few bits of evidence that we have to ultimately test these kinds of models on ecologically and evolutionarily relevant timescales. If we think about size changes over a large temperature range, like people have seen in the fossil record, we would predict that small things will get smaller, large things will get small even more as warming goes up, but all of them will drop their body size in very strong correspondence to the temperature sensitivity, and that can span a very wide range. So the point here for, I think, a SAFS audience is that it's very hard to draw definitive conclusions from a single species, because it can be doing what it's doing because of either its body size, its allometric exponent, or its temperature sensitivity. And one has to I think, look at many, many species to start seeing these patterns. But hopefully this model that I've just presented will help with that kind of generalization. And I also hope that it begins to resolve this, this debate that's in the community and in the literature about the role of oxygen limitation. I think the metabolic index, which is just a measure of how oxygen limitation varies with temperature and can be empirically calibrated, does support the role of oxygen limitation as a driving cause of the observed size responses in rearing experiments, because we can predict those responses using nothing other than measures of oxygen limitation. It also does, so Camp 1 would perhaps shout yay, they were right. The oxygen limitation underlies body size reductions amid warming. Camp 2 can also claim some important correctness, because as one gets toward larger and larger body size, that allometric exponent does come to be very size independent. Epsilon converges on 0, and that does mean that other constraints on body size are going to come to bear, because once epsilon reaches truly 0, there's no body size change of any kind that's going to save you from a loss of aerobic scope. Okay, the model predicts that the degree of temperature size effect has a strong dependence itself on body size, with larger animals, larger species responding more strongly than smaller ones. Of course, that's contingent on them having the same temperature sensitivity, or E0, which is not at all easy to generalize about. Okay, future directions. I think there's a lot of cool stuff to be done here. Testing the model against the observations in the fossil record, including evolutionary terms, which fall out of the math, but I didn't show you, that we could calibrate if we had multi-generational data on respirometry and looked at how the traits vary as these rearing experiments subject more and more generations to changing temperatures. So with that, I think I've used up too much of your time already. I just want to acknowledge again my collaborators and funding agencies, and take as many of your questions as we have time for. Thank you.
Thanks so much, Curtis. That was fantastic. So you've got at least one question in the chat already, which I can read out to you if you'd like.
That would be great. I can't see it.
Okay. It is from Trevor Branch, and he says,
Hi,
it's
Trevor.
from the beginning of your talk. He says, do they get smaller when you cool the temperature too? i.e., is it a warming effect or the effect of taking them out of their comfortable temperature range?
Okay, so I think
and Trevor, if you want to add anything, I can unmute you.
I imagine you're thinking about the experimental data here. And my understanding is that most of these experiments are nominally done with under acclimated temperatures for the initial population, or the initial rearing, the initial rearing would be done at the temperature that they were extracted from. I don't know how closely that rule is followed in these experimental studies, partly because I'm dealing with data that's already been compiled to a substantial degree by previous efforts. The model would, of course, predict that you could get either kinds of response, increase or decrease. And one thing I didn't mention is that these species that we do see actually getting bigger with an increase in temperature, that tail of the distribution, if I can go back to it, up here, that the tail of that distribution where we see things getting larger with warming is predicted by the distribution of temperature sensitivity, E0, because it also has a tail at negative values. So there are some species that show an increasing hypoxia tolerance under warmer water. It's a relatively small fraction of them, but those are the ones that can give rise to an increase in size under warmer conditions. Of course, you could also have the species with if you had a positive epsilon, if larger was better, in terms of hypoxia tolerance, you could also generate a tail of the distribution that would be consistent with this out here. I hope that answers your question, Trevor.
Trevor, if it doesn't, you can raise your hand and I can unmute you. There we go. You're unmuted, Trevor.
I guess I was kind of asking a slightly different question, which is,
Okay.
what if, because all these studies are done with regular temperature and then higher temperature, but what if all these organisms have an optimal temperature range? And so by going outside their temperature range, whether it's increased or decreased, they would have smaller body size. So has anybody ever done experiments of the regular temperature, lower temperature, and higher temperature?
Yes, so that, yeah, I see now that I did misunderstand that question. Thanks for clarifying. I'm on a good slide to answer it, I think, as best I can. You know, here are each one of these little bricks in the wall here comes from an actual set of data that looks like this. So the temperatures serve, the rearing temperatures will span sometimes a wide-ish range. I think on average it's about 15 degrees. And sometimes sometimes it's less, and sometimes you see something that does not show anything like an optimum, right? It's just a straight up decrease in body size. But sometimes you do see things that begin to look like what you're describing, Trevor, which is, it's not quite there. There are other species that show it a little bit more clearly, but you can imagine that if you went toward lower and lower temperatures, these data might start to bend over. They've clearly stopped. You know, they're clearly not decreasing temperature right off the bat. They start doing that after temperature reaches some value. There are one can incorporate that into this model. What it comes down to is that this expression here, E naught, here is probably not a single number for all temperatures. In fact, if you just compute, let's see, go back to here. If we just split these estimates of E naught into estimates that are made warmer than 15 degrees and colder than 15 degrees, we see that there's a statistically significant difference with E naughts being lower on the cold side and higher on the warm side. So that is consistent with this kind of picture. Now, it doesn't tell us yet whether it really would bend over, but the temperature size effect on the cold side is indeed lower than the temperature size effect on the warm side, almost like you would get if you had an optimum that just hadn't quite been sampled well. I think more work would be needed on the rearing experiments and to see whether, you know, really how well that corresponds. Is that closer to what you were asking?
Thank you. Yeah, perfect.
Good.
Awesome. And there's another question from Jim Thorson. I'll just read it again, and then Jim, if you'd like to add anything, just raise your hand and I can unmute you. It says, I believe the derivation was based on rearing experiments. How much do these patterns replicate the observed patterns between temperature and size at maturity for measured in the field for multiple stocks within well-studied species?
Yeah, okay, good question. There would be probably a pretty big mismatch between the size of organisms that are studied in these rearing experiments and the size of the, you know, of organisms in the well-studied stocks that you're referring to. You know, so all these are basically like zooplankton. I'll show you one slide that's from work in progress beyond this, and that is for extending this model to predict growth rates. In fact, one can predict the von Bertalanffy curves from these physiological traits. And I've done that here for Atlantic cod because we have the various metabolic index parameters for Atlantic cod. And you can predict what the von Bertalanffy curve, it's not actually a von Bertalanffy curve, it just has that shape. It's just integrating body size over time from metabolic index. And it predicts those kind of logistic curves, right, where you get, you mature at higher body size for lower temperature, lower body size for higher temperature. It looks like there's hints of that in the data. So with temperatures from different time periods here, in particular focus on the 1980s versus the 2000s, that's the strongest contrast. Colder in the 80s, warmer in the 2000s, of course. The NOAA survey data with otolith aging and body size measurements does support a response that looks like what this model would predict. That's as close as I can come to answering your question, Jim. I think it's an area that would deserve a lot more exploration. I'm partly limited here by the small number of species for which we can actually predict what the growth age curves are from having complete enough physiological traits. That would be one of the data barriers that would have to be overcome to do this for more fish stocks in the field.
Wonderful. You got a very cool thanks from Jim, so I think you answered his question.
Thanks,
I'm
Jim.
not seeing any other raised hands or questions, and we were a little bit over. So I think we should thank Curtis very much. You're getting lots of virtual applause, even though you can't see them.
Thank you.
And I want to invite everybody to join us next week for Dan Ovando, who's going to talk about predictive modeling in fisheries, I believe. So thanks so much, Curtis.
Have a great weekend, everyone.
You as well.